Hello everyone, welcome to the another video lecture of Easy Life Science. Here I am talking about some important questions, their answers with detailed explanation for set exam subject Life Science part 7. So please subscribe my channel Easy Life Science, like my video, share it to your friends and don't forget to press the bell icon so you will get the notification of my newly uploaded video. So let's start. Question first, match the given vitamins in a column first with the related protein in a column second. In a column first, some vitamins are given, and in a column second, their related proteins are given. In a column first, the first option is vitamin C. The vitamin C is known as ascorbic acid. It is an essential cofactor for enzyme prolyl hydroxylase. Deficiency of vitamin C results in accumulation of abnormal collagen because the vitamin C increases the secretion of mature collagen by promoting the activity of enzyme prolyl hydroxylase. So option A, that is vitamin C, matches to the prolyl hydroxylase. Then option B, vitamin K. The vitamin K maintains the healthy blood clotting and prevent the excessive bleeding and a hemorrhage. Vitamin K is necessary for a carboxylation of a protein which governs the conversion of a blood clotting agent prothrombin to the thrombin. So vitamin K is responsible for a thrombin. Vitamin A is a precursor of rhodopsin. The rhodopsin is a photopigment found in the rods within the retina of our eye that help us to see at night. Deficiency of vitamin A causes a night blindness. Rhodopsin is a chromoprotein. It is made up of opsin. The opsin is a colorless protein. So vitamin A matches with rhodopsin. Then vitamin B1. The vitamin B1 is known as a thiamine. It is a cofactor for pyruvate dehydrogenase. The pyruvate dehydrogenase is an essential enzyme for aerobic metabolism. In the absence of thiamine, the conversion of pyruvate to acetyl coenzyme A is inhibited and pyruvate cannot enter the Krebs cycle. So the vitamin B1 is related with pyruvate dehydrogenase. Vitamin C to the prolyl hydroxylase, vitamin K to the thrombin, vitamin A to the rhodopsin, and vitamin B1 to the pyruvate dehydrogenase. Such combination is in option A. So answer of this question is option A. Then the next question, match the following terms in a column first with the appropriate expression in a column second. In a column first, A is a plysiosome. The plysiosome is a large RNA protein complex that catalyzes the removal of introns from the nuclear pre-mRNA. The plysiosome precisely removes the intron from pre-mRNA to generate a mature mRNA, and this process is known as a pre-mRNA splicing. The plysiosome is essential for a cell function and for a post-transcriptional modification. Then the peroxisomes. The peroxisomes are responsible for beta oxidation of long chain fatty acids. The long chain fatty acid contains more than 20 carbon atoms. Peroxisome plays an important role in lipid metabolism, biosynthesis of ether phospholipid, and reactive oxygen species metabolism. In a cooperation with a mitochondria, peroxisome play an important role in a fatty acid oxidation and a fatty acid production. Then the lysosome. The lysosomes are capable of digesting nucleic acid, polysaccharides, fats, and proteins. Lysosome digests or damage its own cells by its own enzyme, which leads to a cell death. The process is known as autolysis. Hence, the lysosomes are called as a suicidal bags or a cell scavengers. Then a centrosome. The centrosome are made up of microtubules. The microtubule in a most cell extend outward from a microtubule organization center in which the minus end of a microtubules are anchored. In animal cells, the major microtubule organizing center is a centrosome, which is located adjacent to the nucleus near to the center of a interphase. The centrosome consists of two microtubule based centrioles, a mother and a daughter centriole. The mother and a daughter centriole differs in age and but they are similar to their structures. So the centrosomes are associated with a microtubules. Plasmosome for a post-transcriptional modification, perioxysomes for a metabolism of long-chain fatty acids, lysosome as a cell scavengers, and the centrosomes, microtubules. So this combination is in option B. Option B is the answer of this question. Then moving to the next question, match the respective behaviors given in a column first with the appropriate explanation in a column second. The first, that is a agnostic behavior. It is a aggressive behavior of an individual within a population, so it matches with a third in the column second. The B, hierarchical behavior. The hierarchical behavior is a existence of a different levels of a dominance within a population, so it matches with a fourth in a column second. Altruistic behavior. The individual behavior where a fitness of a other increases at the expense of a self, it matches with a first. Then a cooperative behavior. Individual contribute toward the enhancement of a mutual fitness, and this combination occurs in option B. So option B is the answer of this question. Then moving to the next question, which one of the following animal possesses a two-chambered heart? The answer of this question from the option is a goldfish. The goldfish have a two-chambered heart, whereas the amphibia and most reptiles have three-chambered hearts, two arteria and one ventricle. Mammals and birds have a four-chambered hearts with two ventricles and two arteria. Frog, crocodile, ostrich, pigeon, bat and whale have a four-chambered heart. So answer of this question is option C, that is a goldfish has a two-chambered heart. Then moving to the next question, in eukaryotic chromatin organization, which one of the histone seals off the nucleosome at the location at which linker DNA enters and leaves the nucleosome? The H1 histone protein is one of the protein in a 5 main histone proteins which are a component of a chromatin in a eukaryotes. The H1 protein binds to a linker DNA. So here question is asked, which of the histone is seals off the nucleosome? Answer of this question is H1 histone is responsible for seals off of the nucleosome. Then moving to the next question, which one of the following parasites do not cause a lymphatic filariasis? Brugia malayi, Brugia timuri and Ochea bronchopti. These three organisms are responsible for lymphatic filariasis, but the Mansoniella streptocerca is not a responsible parasite for the lymphatic filariasis. So option D is the answer of this question. The next question, a species adapted to a specific habitat is known as. A species which is adapted to a specific habitat is known as a ecotype or a ecospecies. A ecotype or a ecospecies describe a genetically distinct geographic variety, population or a race within a species which genetically adapted to a specific environmental conditions. Then the other options like a biome. The biome is a biogeographically unit consisting of a biological community that has formed in a response to the physical environment. It is broader term than the habitat and can comprise a variety of habitats. The ecotone are an area of a steep transition between ecological communities and ecosystems. For example, marshland is an example of a ecotone. The marshland is a steep transition between the dry and a wet ecosystem. Another example of a ecotone is a mangroves forest. This is a steep transition between a terrestrial and a marine ecosystem. Then a grassland ecotone, it is a transition in between desert and a forest ecosystem. Estuaries are also example of a ecotone in between a salt water and a fresh water. And the niche describe the role of an organism plays in a community. A species niche encompasses between the physical and environmental condition that required by the species like a temperature and a terrain, and its interaction to the other species like a predation or a competition. So for this question, a species adapted to a specific habitat is known as a ecotype. Then the next, the phylum that has characterized by a animals which have a water vascular system is. Here the question is asked on a phylum, the phylum Echinodermata, and their species are known as having a water vascular system. Water vascular system is a hydraulic system used by echinoderms such as sea stars and sea urchins for locomotion, food and waste transportation, and respiration. This system is composed of canals connecting the numerous tube pits. So the answer of this question is echinodermata. Then the next question, the finches on Galapagos Islands have a widely differing beaks adapted according to their diets, and this is an example of what? Adaptive gradation, parallel evolution, co-adaptation, and adaptive convergence. The finches on Galapagos Island have widely differing beaks adapted according to their diet. This is an example of adaptive gradation. Adaptive gradation is a process in which organisms diversify rapidly from an ancestral species into a multitude of new forms, particularly when a change in the environment makes new resources available, alters a biotic interaction, or opens new environmental niche. So this is an example of adaptive radiations. Here in this image from one ancestral finch, a number of other types of finches are adapted. The vegetarian tree finch, large insectivorous tree finch, small insectivorous tree finch, woodpecker finch, warbler finch, cactus ground finch, sharp-beaked ground finch, small ground finch, medium ground finch, and large ground finch, and they are playing a different role by using their different beaks. So this is an example of adaptive gradation. The other option like parallel evolution. The parallel evolution occurs when independent species acquire a similar characteristics while evolving together at the same time in the same ecopace, whereas the co-adaptation refers the mutual adaptation of a species which depends upon each other. So for this question, answer is the adaptive radiation. Then the next question, which one of the following animal is named as a living fossil? Where the animal is persisting above 400 million years without further major morphological evolution. The answer of it is horseshoe crabs. The horseshoe crabs are a living fossils, meaning they are existed or they have existed nearly unchanged for at least 445 million years back. Horseshoe crabs are not actually crabs, they are much more closely related to the spiders and other arachnoids than they are to a crabs or a lobsters. So answer of this question is a horseshoe crabs. The cyanobacteria are also considered as a living fossils, emerging from last 3.5 billion years ago. They exist as a single bacteria but are most often pictured as a stromatoliths. Stromatoliths means artificial rocks produced by a cyanobacterial waste. Moving to the next question, which one of the following factors does not contribute to the allopatric fasciation? The allopatric fasciation occurs when a species separate into the two separate groups, which are isolated from one another. A physical barrier such as a mountain range or a waterway makes it impossible for them to breed with one another. So, allopatric fasciation prevents a gene flow. The option A, the isolated population is exposed to different selection pressure as compared to the parent population. Yes, this is true for allopatric fasciation. A population becomes geographically isolated from a parent population. Yes, it is also true. Option C, there is a gene flow between the two separated population. It is false. There is no any gene flow when the two species are separated through a barrier or when they get allopatrically fasciated. The gene flow is the movement of alleles from two species. It is relatively free because individuals can move and then mate with individuals in their new location. In a sympatric fasciation, the groups from a same ancestral population evolves into a separate species without any geographical separation. So, here in this image, the sympatric fasciation, a definition is given. A new species evolves from a single ancestral species while living in a same geographical niche. Here in this one park, the two fruits are developed which have a different color and they are evolved from a one single species but having in a one geographical niche. So this type of a fasciation is known as a sympatric fasciation. Then the parapatric fasciation. In a parapatric fasciation, new species evolves as a result of a partial geographical isolation as a result of occupying a new or a different niche. Allopatric fasciation, when the species from one population goes to another region and develops or evolves there and forms a new species and formed a new species and original species does not mate with each other and they are separated by a geographical barrier, and this type of fasciation known as allopatric fasciation. So for this question, answer is the option C. Then next, match the hormones in a column first to their respective physiological effects in a column second. Melatonin. The melatonin is a hormone which is responsible for a circadian rhythm. In animals, melatonin plays an important role in the regulation of sleep and wake cycle. whereas the oxytocin is a peptide hormone and it is a neuropeptide normally produced in a hypothalamus and released by a posterior pituitary gland. The oxytocin plays a role in a social bonding, reproduction, childbirth. The oxytocin hormone is released into a blood as a hormone in response to a sexual activity and during the labor. Oxytocin stimulates the uterine contraction and to speed up the process of a childbirth. It also plays a role in a bonding with the baby and a milk production. So melatonin, a regulation of circadian rhythm, oxytocin, secretion of milk and uterine contraction. Then the cholecystokinin. The cholecystokinin is the hormone that functions as a part of our digestive system. It is released by a small intestine during the digestion. It is sometimes called as a pancreozymin. Cholecystokinin stimulates the gallbladder to contract and release stored bile into the intestine. So its function, that is a secretion of an enzyme from a pancreatic acinar cells. Then a prolactin. A prolactin is a hormone made by the pituitary gland. Prolactin causes the breast to grow and make a milk during the pregnancy and after birth. So for prolactin, secretion of milk in a mammary glands. The somatostatin, it is a cyclic peptide well known for its strong regulatory effect throughout the body. It is also known as a growth inhibiting hormone. It is produced by a gastrointestinal tract, pancreas, hypothalamus and a central nervous system. So somatostatin, it is known as a inhibition of or known for inhibition of a secretion of a growth hormone. So A for 5 or A matches to the 5, B with 3, 5, 3, 4, 2, 1. This combination is option D. Option D is the answer of this question. Then moving to the next question. Which one of the following hormone initiate a signaling cascade by directly binding to a intracellular receptor? Read this question carefully. The hormone which have a intracellular receptor. The steroid hormones which are lipid soluble and can easily pass through a cell membrane of the target cell and reach into a cytoplasm. The hormones like aldosterone, cortisol, estradiol and a progesterone are steroid hormones and pass from a cell membrane of a target cell and acts on a intracellular receptor. The intracellular receptor are globular proteins located inside the cells rather than its cell membrane. Out of the 4 options, insulin, gonadotropin and epinephrine, these 3 hormones have a receptor on a cell membrane, whereas a progesterone is a lipid soluble hormone. It has a intracellular receptors. So answer is a progesterone. Then next, which one of the following bond is not present in a ATP? ATP is a adenosine triphosphate. It consists of a nitrogen base, that is adenine, pentose sugar, that is ribose sugar and a 3-phosphate molecule. In between sugar and adenine nitrogen base, there is a bond, that is a N-glycosidic bond and forms adenosine. The adenosine joins with one inorganic phosphate through a phosphoester bond and forms adenosine monophosphate. This adenosine monophosphate further joins to another inorganic phosphate through a phosphoanhydride bond. But in ATP, there is no such evidence for a alpha glycosidic bond. Phosphodiester bond is there, phosphoanhydride bond is there, N glycosidic bond is there, but alpha glycosidic bond is not present in a ATP. So answer is the alpha glycosidic. The reaction involved in a direct conversion of L-phenylalanine to a L-tyrosine is. The answer of this question is a hydroxylation. The phenylketonuria is a genetic disorder caused by a defect in a gene coded for the enzyme phenylalanine hydroxylase. Body uses this enzyme to convert phenylalanine into the tyrosine. The tyrosine is necessary for the production of a neurotransmitter such as epinephrine and a norepinephrine and a dopamine. If a phenylalanine hydroxylase is not produced due to a defect in a gene, then the body cannot break down a phenylalanine and causing it to build up in the body. High level of a phenylalanine can saturate the transport system across the blood to brain, and it leads to finally brain damage. So this is known as a phenylketonuria, and here there is a lacking of enzyme phenylalanine hydroxylase. So in a conversion of a L-phenylalanine to a L-tyrosine, the reaction involved is a hydroxylation through a enzyme phenylalanine hydroxylase. So answer of this question is a hydroxylation. Then the next question, the human major histocompatibility complex or a MHC complex is, it is polygenic and a polymorphic. MHC is a polygenic. It contains several different MHC first and MHC second genes, so that every individual possesses a set of MHC molecules with different ranges of a peptide binding specificity. Presence of different alleles of genes at a frequency more than 1% is known as a polymorphism. MHC is a polymorphic. MHC gene shows a polymorphism in a human genome. So answer of this question is a polygenic and a polymorphic. Then moving to the next question, binding of an antibody to its cognate antigen does not involve. Here the bonds in between antigen and antibody are asked. In between antigen and antibody, we can see for electrostatic interaction, hydrogen bonds, van der Waals forces, hydrophobic interactions. But there is no covalent bond in between antigen and antibody. So the answer of this question is a covalent bond. Then the next question, a globule protein of a molecular weight 50 kilodalton exists as a mixture of a monomers and a dimers in a solution. The most appropriate technique for a separation of these two forms of a protein is. Here the question is asked for an analysis of a protein by a technique. For this purpose, we can make a use of a gel filtration chromatography. A gel filtration chromatography separates the proteins on the basis of their molecule size. The separation is achieved by using a porous matrix to which the molecules have a different degree of access. That is, a smaller molecule have a greater access and larger molecules are excluded from the matrix, and hence they elute first. So for analysis of a globule protein or a separation of a globule protein, the technique used is a gel filtration chromatography. So thank you for watching me. So please subscribe my channel Easy Life Science, like my video, share it to your friends, and don't forget to press the bell icon.